Are recording, so no more profanity. Um, well, it's certainly possible to graph things. There really is no holistic way to compare the two, but we can look at features of each of the collections. So the one we used our last class meeting, the collection of body weights in this room and that room, what was the primary feature that we came up with? We took two body weights out of this room, and then we characterized our entire room by its, not nice and happy, but what's the opposite? Mean. So we can take the mean values, and then what we're doing in our testing is comparing that distance. And there are alternative measurements. We can use the variability of things as a measurement. We could use the skew, we could use a whole bunch of our descriptive statistics, and we can make measurements up. So for example, the distance between the two is itself a measurement. And if we can convince ourselves that that distance between the two is normally distributed, then we have a whole set of tests which were called, had the word metric in it, but was it parachute metrics or walking metrics, or we had parametric tools to evaluate that distance. So step one, we have two collections. Step two, we come up with some measurement from each of the collections that we're going to ask the question of. So if A and B were the same in that drawing right there, what value would you expect that distance to have? Big or little? She's making a circle with her hand, so we'll go 0 is about as small as you can get when we're talking distances. You are correct. And as that distance gets big, they're different. And at some point, we declare how big is too big. And when we do the how big is too big, that used a Greek letter of the alphabet. And does anyone remember what that Greek letter is? It's the first one. It's not omega. It's alpha. Which essentially was a measure of overlap. And so we can control how much overlap we're willing to accept to declare them the same or different. So in general practice, that overlap is usually set at either 2.5 or 5%, depending on whether our cat here has 1 or 2 tails. So if we care about either side, if we don't care about which side the distance is, then it's a two-tailed test, and we would still have 5%, but be 2.5% on each side. So generally, if that overlap is, that's right, smaller than our prescribed alpha value, which is usually 5%, we declare them different. We reject the null hypothesis, which by default is almost always the same. And if the overlap is bigger than that, we do not have sufficient evidence to declare them different. And that's the essence of a hypothesis test. Now I chose the letters A and B not entirely arbitrarily, because today we're going to examine another interpretation of this testing scheme. So A and B can represent differences in collections based on some treatment that we apply. So A could be everybody in this room, B could be all the invisible people after we've done some treatment to them, like ridicule them when they're not here so they can't hear us, although it's being recorded right now. So all you out there in TV land, you're being ridiculed right now. You should see the hand signs everyone's here is making. It's funny. And that in modern terminology, it's called A and B testing. And so A can be unvaccinated, B can be vaccinated. Like I said, it's probably a really politically poor choice to make nowadays, but it's in the news. So, and the question we want to ask, is the B group different than the A group, And more importantly, is the vaccine the cause of the difference? And so we're not going to just see if they're red or blue, but we'll look at some deeper aspects of that. And so that deeper aspects might be the number of hospital admissions, and so forth. And we want to pose that question because if the treatment, using that example, if the vaccine really makes no difference, it's a waste. There's no point in administering it. And that's what they do at the Centers for Disease Control right now as we speak. They're sitting there doing A and B testing on something. Probably not everyone. They're probably mostly just administrators. But there's some poor sucker there who, in between emptying the trash cans, has to do data science for them. Your government at work. Ooh, that was recorded. Watch my mouth. Okay, so let us go to, oh, it's going to be one of those days, isn't it? Let's see a really backward way to get to our, there we go. And I actually want to go to the home computer. So if you ever go this way, you just go down to teaching, and this is what we're used to. But I want to run it on the development computer. Ooh, I got risk ahead. That is risky when it remembers your password. Well, there you go. So today is, I guess it's lesson 17. All right, so the formal title is Testing for Treatment Effects. And so it's going to build on what we had last time. All right, Catherine, Mark, and Mills. Joshua, can you see that back there? Good enough. I mean, I know you're watching YouTube videos on the side, and that's fine. So here's what we're looking at today. Testing for treatment effects. If you recall from last time, hypothesis testing was a procedure to systematically decide if two collections are similar or different by comparing descriptive statistics. Typically, the comparisons are made on means and variances or linear combinations of those. There are other measures that can be compared, but when we get to the higher moment measures, that's outside the scope of this class. The underlying technology is the same. You make the measurement, you run a simulation to see how the measurement should behave, and then you make your decisions based on those results. I'm using an example from this book, which is formally this is the textbook for this class, but I'm operating under an assumption, but we could test that right now, that probably nobody has ever visited it. Which is fairly standard in university courses. So it's all in testing hypothesis, and it's kind of a cool book until you actually try to run any of the examples. Because nowhere in this book does it tell you that you have to load the data science external package. Nowhere in the book. It does use the word import at some place, and then nowhere in the book does it tell you how to install that thing. So they have an example on jury selection. We'll skip that. We're going to do the example on Mendels models. So that is where what we're going to do comes from. Okay, so let's get back to ours. So in that book, there's a secret module. It's not secret, but that you need to install, and the book tells you no way how to do it. So welcome to the world that a few of you are computer scientists in this room. A lot of invisible people are. You're going to be taught indirectly a technique called obfuscation. This is a good example of it. Everything's open source, we're sharing it with you, but oh yeah, there's this secret agent decoder ring that we didn't bother to mention. In the case of this particular textbook and the original authors, that's probably just a screw up on their part. It never occurred to them that nobody would know not to know how to install it, because it was designed for a class at the University of California, Berkeley. Okay, so enough of my complaining. What I'm demonstrating now, this command up here is how I installed it. I think in your guys' environments, if you use the exclamation point, keyword pip, and install data science, it'll go out to the internet and attempt to get it and install it for you. If that doesn't work, you'll have to go to the Anaconda PowerShell prompt, and again, get into the correct path for your Anaconda install, which is usually something like optconda. It again depends on your particular computer, and run the install. You only have to do it once, and once it's installed in your local environment, it's there. So I've done it on the server environment, and so I'll go ahead and install it in a second. I'm also going to install NumPy as NP, matplot library, pyplot as PLT, and then for a reason I don't understand on my computer, I have to put the magic function matplot inline for it to draw graphs. And it's the first time I've had to do that in this class, so I don't know what's broken, and that should be get. So let me do those preliminaries because that's not important for the lecture per se. So now we're going to go look at Mendels Pea Flowers. So some of you probably have heard of Gregor Mendel in a biology class. He is the cause of the entire chapter on genetics in biology, and had he never done any of his work, we wouldn't have to worry about it. And he is the author of the famous book, I Got It From My Mama. And there is a picture of the book cover. He didn't write that, someone else did. That's a book for kids to explain how features are passed from parent to child. The picture on the right is the, quote, large-scale research station he used. That's the rose garden in the back of a monastery in Austria. And I didn't take that picture. I've never been to that one, but I've been to Austria, and they do have gardens like this. The place is just lousy littered with them. And they make great parking lots, a good place to put a 7-Eleven or a Stripes, and they're wasting it growing plants. So I just don't get it. So Mendel is noted for performing large experiments on plants to come up with the fundamental laws of genetics. Something tells me he could care less about genetics. He was trying to grow plants to make some kind of wine or sell them at the local market. And he planted a lot of pea vines. And if you're not familiar with them, the flowers on a pea vine are actually quite pretty if you're into that. And he was trying to get either purple or white, and I'm going to guess that in those days the purple was the target plant that was desirable. And he hypothesized that the plants should produce purple and white at roughly a ratio of 3-to-1. Now that's given him a lot of credit. There's no way he hypothesized that up front. He watched it through 8 or 10 seasons and noticed that, yeah, I get about 3 times as many purples as I do whites. And then he got into mathematics, because when you're stuck alone in an Austrian monastery with nothing but Benedictine and brandy to live on during the winter months, your mind kind of wanders. He could only do Gregorian chants for so long, so he had to think of something else. So Mendel's model, translated to our way of thinking, is that for any plant, there's a 75% chance it'll produce a purple flower, 25% chance it will be white, and it's regardless of all the other flowers around it. So that last part, that regardless of, is very similar to a feature we mentioned last time called, well, regardless is dependent or independent? Independent. And identically distributed. So let's go ahead and let's decide if we think Mendel's model is a piece of crap or not. We have a tool that he didn't have. If he had an Apple computer, he never would have gotten off the Benedictine and brandy. And we can simulate the behavior of many simulated plantings and growings under the assumptions that the model is correct, and then see what that would be expected to produce in a statistical or a probabilistic sense. Essentially, we simulate growing 1,000 plants and count how many come up purple, how many come up white. We do that in computer. Then we do it again. Then we do it again. And we keep doing it again until we get blue in the face, maybe 10,000 times. And now we have 10,000 simulated experiments, and we can study the behavior of those. We can ask ourselves, did we indeed get roughly 3/4 purple and 1/4 white or not? And our goal is to see whether or not Mendel's model is good. Now, in the case of Mendel's model, we're going to have his actual experiments. So his actual experiments won't come out exactly 3-1. In fact, if they did, one would be very suspicious. But we'll have our simulated group. We have his one actual experiment, and we'll ask the question, is the distance from the actual experiment to the simulated measurement too big or not? That's the essence of what's going on here. So our statistic, or think of it as our measurement, is going to be distance, and it's going to be the fraction of the sample that's purple minus the fraction of flowering plants, so it's purple minus the total minus 75%. And that would represent a distance. And so if we have 75% purple, I think that's supposed to be a divided by. Yeah, that minus is supposed to be, I'm going to fix that. That's what happens when you copy other people's work. Purple divided by flowering would give me a fraction. And let's, since we have time, we'll go ahead and make it actually pretty. There we go. That's nice. I like that much better. So we have the fraction of purple in our sample minus 75%. So if our sample is indeed 75%, what's the distance there? 75 minus 75 is, you can go ahead and make the hand sign. 0. So that is our measurement. So when you take your probability and statistics class, if you haven't already, don't fight with the instructor because you'll lose, but whenever they say statistic, you can think of some kind of measurement, because that's all it really is. So now let's predict what the measurement would be if the model is correct. And that's key to coming up with how to build hypothesis tests. So to see how big the distance would be if Mendel's model were true, we can use from the data science package a function called sample proportion. So this part's pretty cool because somebody's done the work to get proportions for us to simulate the distance under the assumptions that the model's true. First, we got to figure out how many times to sample, and to do that, remember, we're going to compare a simulation with Mendel's actual experiments. So we should simulate the same number of plants that he had. Mendel grew a lot of plants. So in one of his experiments, he grew 929 plants, which is a weird number if you ask me. So he probably grew 1,000, and 70 of them just died of the variety corresponding to the model. So we have to sample 929 times. And then doing that will generate one value of our measurement. So the steps in the simulation calculation are to sample 929 times at random from the distribution prescribed by the model, and then find the sample proportion in the purple flowering category, multiply that by 100 to get a percent, subtract from that percent 75 and take the absolute value of the difference, and that becomes our measurement. So because we have absolutely no naming creativity, we're going to create a function called distance from 75, and it takes an input parameter of p, which represents the proportion of interest, and then it randomly returns the absolute value of 100 times p minus 75. So to simulate one value of the distance, first we have to simulate the proportions of purple flowers among 929 plants under the assumptions of the model, and then calculate the discrepancy. I guess I should run these cells as I go, otherwise when I get to the end the plots won't look very good because they'll just won't work. So here is a object called model proportions. What kind of object is that? Python question. It is a list. Has how many elements? One of them is, and the other one is 0.25. So the sum of the elements of the list adds up to 1. So it's just a bucket with either purples or whites in it, and if you pull out a 0.75, you pull the purple. And in time-saving things, you could have made it purple and white and then had some other manipulations to get it into numbers. And now we're going to use the built-in function sample proportions, and we're going to create an object called proportion purple in a sample. We're going to run the sample proportion function, which samples from our list named model proportion 929 times, takes the first item in the result and places that to the proportion of purples in the sample. And then we run our distance from 75 function to proportion of purple in the sample. And when we do that, in this particular instance, we got a distance of 1.74. So that was one simulated sample. We're going to repeat that a bunch of times so we can generate a probabilistic behavior. So to get a sense on the variation of distance, we got to run the simulation over and over and over again. And that's what real data scientists and statisticians do in order to make a page of a textbook that you can't read. So in this case, we'll run it 10,000 times. First, we have to do the single instance, and so we're going to create a function called one simulated distance, not very creative. And what one simulation distance is going to do is just return the distance in a single sample event. So we'll get that defined. And then we'll use our friend the for loop to do 10,000 simulations. So what it's going to do, it'll do one simulated distance. It'll append to an array named distances the result of that. And when we get done, we should have 10,000 things. So we let it run, and we got this 10,000. Now you've probably noticed in your own scripts, if you care, that you might decide that that's an annoying output. And I think I just learned the other day that if I put a semicolon, it will suppress that output. So it still calculated the length of distance, just didn't do anything with it because of the semicolon. We'll see the value of that in a second when we get to graphs. So now that we've made all these simulated distances, let's create our friend the histogram. And on the left-right axis, we're going to plot the value of distances that's in our 10,000 long sample. So we have 10,000 simulated growings. And on the up-down axis, we will, see, I think we will count the number of things in each of the bins. So we have a bin from 0 to, I guess, 0.8 or 0.6, from 0.6 to 1.4. And the binning is kind of goofy because I'm just letting it do it itself. And we have 10 bins. So do you get the idea of what the bins are? The computer is assigning the width of the bins so that the total collection adds up to 10 of those. And we can see that in values of 5 and 6 and beyond, there's not a whole lot of things in the bins. If we wanted to do a different value, we could do, see, 10 gets me there. Let's see what 20 does. I may regret this, but. So there is our, yeah, it just does not come out on a whole number very good. Yeah, I'm not going to get any better now. Try 11. Maybe I can at least get it. Okay, those are close. Those bins are close to 1/2 unit width. There are ways to control the width of the bin, but I'd actually have to read the instructions, and that's not going to happen because I'm a professional engineer. So there is our histogram of distances from a simulation model that by design follows the actual model proportions. So we designed this so that we should 3/4 of the time get purple, 1/4 of the time get white. Therefore, we should get distances of 0 every time. But because of random variation that happens like it would in real life, we get distances that range from 0 here up to about 5. So 5 units we might conclude, that's kind of far away from the 0. So if we did an actual experiment and we calculated a distance of 5 from the 75th percentile, we might conclude it's probably not following the model. I mean, we have evidence here that it could be, but the chances of it doing are, like 10 out of 10,000. That's not very good odds. But if we do that same thing and we get a distance of 0.4, for instance, that's clearly in the first bin. That's 300 out of 10,000. That's 30%. 30% chance that we would see that if indeed the model was true. We can play more games. We have a function in NumPy called Quantile, and we can look at the distances array and calculate the 25th, 50th, 75th percentile and the 99th percentile. We're doing this manually. We're doing something similar to a feature that's available in Pandas. These aren't data frames yet, so we don't have the Pandas tools available to us. We could attempt to use what function to get the 5-number summary. So we're doing 4 of the 5-number summaries. What's it called? Describe. Yeah, if we put describe on that object, we'll get a red rectangle of encouragement, but we won't get what we want. And that's because distances are not a data frame. So we could just use Pandas data frame and convert it to a data frame, or because we don't care that much, we'll just use the Quantile function in NumPy. So we're going to compute these 4 quantiles, and then we'll print them out back to the screen. And we do that, and it says that 25% of the distances are smaller than 0.46. That's probably what I should have typed there. 50% are smaller than 0.94. 75% are smaller than 1.64, and 99% are smaller than 3.63. So almost all the distances are smaller than 4. So if we were to calculate our own statistic on a real experiment, and we got a distance bigger than 4, what's the likelihood that observation came from this model? Low or high? Low. Good. On the other hand, if we got a distance of 0.1, we could comfortably conclude it's a pretty good chance that would happen, because the 99th percentile is everything less than 4. The 25th, that last category, is everything less than a half. So if we get 0.1, we're happy. We can also make the same plot using the data science tool, and pretty much all this does is it lets you put a title and it lets you choose a particular column. So there's hardly arguably much of an advantage to it, other than if you're following the UC Berkeley document, you want to use their scripts. If we look at the horizontal axis, we can see typical values of the distances predicted by the model. They're pretty small. A high proportion of the distances are between 0 and 1, pretty much 50 and 40, almost 95% are over here. In this case, the data science package by default plots the vertical axis as a density. We plotted it as a count. There's different things we could put into the plot function to get the same output. Now let's compare our prediction and the actual data from one of Mendel's experiments. He did an actual experiment where of the 929 plants that grew, 705 were purple flowering. And that is a percentage of 75.89. And so the next question we want to ask ourselves, how expected is that relative to our simulation model, which by design is the way things should behave? So we'll get the observed statistics as 0.89. And if we look at it just by eye, we see 0.89 is less than 1, and we're in the two big columns. So we would be comfortable saying that, yeah, Mendel's experiment agrees with this 3-to-1 ratio according to our simulation. But because we're anal, we're going to go ahead and plot it. And here we're going to take our same histogram, only let me modify that to 11 so it agrees with the upper one, and plot it. And then we're setting the alpha channel of a half so that it has some transparency. And then we're going to do a scatter plot right on top of it. And we're going to plot observed statistic on the x-axis and 0 on the y-axis for lack of, if we don't do that, it'll get mad at us. We'll make it red, and we're going to size it at 150 pixels. And then we will show the plot, and we get that cool picture, and that red dot right there is our test statistics. If you don't like half circles, then plot it at about, see, it's 150, so what's half 150? 75. And it puts our dot right there. So that dot is representing our one actual measurement compared to 10,000 simulated ones. And so I ask you the question, so is that one actual measurement? Are you comfortable with the underlying model? Yes or no? All quiet today. Nobody's saying anything. Yes?
So, like, what value, like the 0.89, like, at what value would we not be comfortable with putting that?
Ah, thank you. That's leading into the next part of the discussion. Let's do the same thing with the data science package. You get the same picture. Okay, so the observed statistic is like a typical distance predicted by the model. So by this measure, the data are consistent with the histogram that we generated, and this is evidence in favor of the model. We can further go on to find the quantile associated with that 0.89. And so let's see what its quantile is relative to the whole 10,000 distances of the sample. And here we're just brute forcing it, so we're going to start at 0, which we know should not be selected, and we're going to test until we get a difference between the observed statistic and a quantile based on that Q value of less than 0.001, so essentially 0, and that will tell us the quantile of the observed statistic. And I'll just run it, and I think this takes a little while. because I had to do 10,000 runs until it found how close was small enough. And so the observed statistic, 0.888, is smaller than 54% of the simulated sample. So that 0.888 is roughly the median value of our simulated sample. So we have to decide, is median close enough or not? But let's just, for grins, let's suppose that our observed statistic came out to be 0.1, because that's pretty close to 0. And we run the same thing and we find out that our observed statistic is smaller than 94%, we would probably be pretty comfortable. And 94% leaves us with a little bit more than 5% overlap error. And that's the essence of how we go ahead and answer the question that you asked. Let me restate that. Okay, so the example overall is the essence of hypothesis test. So let's review the elements of a statistical test as we presented last time and as will be presented in any probability and statistics class that you get. There's 4 essential elements. A null hypothesis, or when the model's correct, an alternative hypothesis, whether we're going to be bigger than, smaller than, or either side, a measurement, it's called the test statistic, and then a rejection region. So let's suppose we wanted to make a test concerning some parameter theta based on some random sample, y1, y2, blah, 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 yn. From that sample, we compute an estimate of the unknown parameter called theta hat, and we'll assume for this lesson that the estimator is approximately normal, distributed with mean 0 and variance of sigma squared theta hat. Excuse me, mean of theta and variance. So we're taking a sample, we're going to compute a measurement from that sample, and we're going to pretend that that measurement is the measurement from the population that we don't know anything about. And suppose that theta 0 is a particular target of theta. So let's pretend that theta 0 happens to be the population value. We want to test our null hypothesis that theta equals theta 0 versus the alternative that theta is bigger than theta 0. So these little distributions here, moving from left to right, represent different realizations of the theta hat parameter. So if our theta hat distribution was that one, it's pretty likely that our theta hat is a correct estimator of theta 0. But as we start moving away from the true value, that chance decreases considerably. So we've got a little overlap here. We have none on that one, none on that one, and so forth. So putting that in words, if theta hat's close to theta 0, it's reasonable to accept the null hypothesis. However, if theta is bigger than theta 0, it's more likely that theta hat is also going to be large. And so the values of theta hat larger than theta sub 0 favor the rejection of the null hypothesis that they're equal, and the acceptance of the hypothesis that theta is bigger than some value theta 0. So using those 4 elements above, we can concisely state the hypothesis test. Null hypothesis, our estimator or our population value is equal to theta 0. Alternative, our population value is bigger than theta 0. Our test statistic is theta hat. It's the measurement made from the sample. We're pretending that the sample behaves like the main population. And the reject region is theta hat. Our measurement is bigger than some value k. And then we have to figure out how to pick k, which was your question. How do you choose k? Excellent question. Don't have an answer just yet. The actual value of the rejection region is determined by setting the type 1 error probability that we want to accept, which is called alpha. So it's called last time, it's still called alpha, and then choose k accordingly. So in this particular example being presented here, it's a one-tailed test, and so we're going to declare that our overlap region out here is alpha, and if we get a test statistic bigger than this value k, that puts us out in this right tail. We're determining that there's not enough overlap to accept the null hypothesis. So typically, again, alpha is set at 5%, and we determine the value of k so that the area from k on out to infinity is 5%, and that's how the value is selected. If the null hypothesis is true, and theta hat has an approximately normal distribution with mean theta 0 and variance sigma squared theta hat, and we desire a type 1 probability error of alpha, then k is equal to theta 0 plus z sub alpha theta alpha hat, where z is just the z-score for this value, mean 0, variance sigma of theta hat. And that's the choice with a probability that z, a realized value, is bigger than z alpha. And that probability is equal to alpha. So basically we're doing the integration using a z-score. So that reject region could further be expressed as theta hat minus theta 0 over sigma theta hat is bigger than z of alpha. And so finally our test becomes theta equals theta 0, theta is bigger than theta 0, our test statistic is written up there, and we reject when the computed statistic is bigger than z sub alpha for some value of alpha. And remember, you get to choose alpha. So now let's see that done by example, because seeing it done in algebra is somewhat useless. So let's suppose we're in a factory, and we have a factory that, unlike modern factories, must be stopped and maintained when the defects exceed 10% on the daily production run. Bear in mind, modern factories, we just go ahead and put those in different packaging and sell them at the dollar store. But back in the olden days, they would shut down the factory and fix the broken machinery. So we are the engineers for the factory, and in between our cups of coffee, we collect a random sample of 100 items from one day of production and find out that we have 15 defective items, and the shop foreman, the guy that actually runs stuff, claims that we've got to shut everything down and fix the machine and repair. And us, the engineer that represents management that doesn't like to shut things down because we perceive we're not making money, says, wait a minute, let's be sure that your claim makes sense. Is indeed 15 defects indicative that the machinery is starting to fail? So let's apply our process. Capital Y will denote the number of defects. And it turns out that Y will have a binomial variate, so it'll take on a value of true or false, 0 or 1. And you wouldn't know that a priori. You'd have to have some probability knowledge on what a binomial is. So it has a chance of defect with probability P and not defect with 1 minus P. So think of it as a coin that's weighed funny. Instead of turning up heads half the time, it turns up heads a tenth of the time. And let's say the defect outcome is the value P. We'll assume 100 is large enough so that we can approximate the binomial distribution with a z-score statistic. So again, this is appealing to your probability and statistics class, which you may or may not have had. If you've not had it, it's just a function. So we have a function that we can evaluate. So our null hypothesis is that the defect rate is 10%. An alternative is it's bigger than 10%. Our test statistic is the z-score based on those proportions shown up there. And our reject region will be z is bigger than z alpha for some value of alpha. And we'll choose alpha of 1% because we're in the manufacturing business. We're making microchips for cell phones, and so we have to have a really low defect rate. Otherwise, we're going to lose out to Huawei or whatever other company is competing with us. And so to evaluate that, we have to do a little bit of scripting. Remember our friend NormDist? Sure you do. He was here last time. So that's just a normal distribution function, hand-coded ourselves instead of using built-in one, which is kind of stupid because there's built-in one. We're going to take a sample of 100. We counted 15 defects. We're going to determine the proportion of defects, and we're going to call that fat or p-hat. And we have p0 of 10%. So that's the design defect rate, and this is an observed defect rate in a single sample. We'll determine the z-score of that, p-hat minus design divided by, that is the sample variance expressed in code, and that gives us our z-score of our test statistic. Next, we want to determine either the z-score of alpha arbitrarily, or I have a note here that says we'll do this by trial and error. What we're going to do is we're going to adjust this number until we can get the z-test and z-alpha equal. And that number, when fed back into a normal distribution, will tell us the quantile, or the area under the normal distribution curve. It will tell us that area there. And if that area is bigger than our desired one, we would reject it or not. And so initially we're going to look at this quantile. We're going to ignore all the rest of it. And once we get the quantile equal to, I think our target was 0.01, then we will interpret what's coming out. So let's go ahead and run that. So I'm starting with a guess of 1.646, and that tells me that the area from 1.646 onward is 0.049, so a little under 5%. If I guess 2 point, that's not what I want to do, Control Z. If I do 2.325, I'm a really good guesser, or I looked at the table of z-scores in the back of my statistics book that was printed back when dinosaurs roamed the earth. I get an alpha of 0.01, which is our target value. And so now we can look at the remaining information. Our test statistic from our observations is 1.667. Our reject value is 2.235. So is 1.667 bigger than 2.235? That'd be a no. So in terms of our picture up here, we're over in this area somewhere. We're not quite far enough to hit the rejection region. So our conclusion is do not reject the null hypothesis. Our type 1 error probability is 1% by design. The p-value, if we were to reject, we would be rejecting at an area of 5%. So we turn back and says to the foreman and say, no, we're not going to shut the line down. We don't have enough evidence that the machinery is failing. And nowadays, what does the foreman do at that point? He says, you can take this job and shove it. I'm working here, and walks out. And then you have a real problem as an engineering manager. You got to have somebody run the machinery. So is it any wonder why we're being replaced with robots? Robots don't get pissy like that. Fortunately, you're in a class, you're going to learn how to drive the robots, you'll be fine. Now let's play the same game and look at the actual defects by simulation. If we do that, it's running. So Here we did 2,000 simulations, and let's see what I set the defect rate as. So using a uniform random number generator, and we are selecting based on a proportion that we supply. So if we put in the theoretical defect rate of 10%, and we do 2,000 simulations, this is our defect rate at 2,000 random simulations that are performed according to what our design model is. And you see that we get 10% a lot of the times, but sometimes we get a little bit more. In this particular instance, we get a few less, and then once we get far away from 10%, the distribution drops off quickly. If we don't think 2,000 is enough, let's go to 3,000. No, let's go to 5,000. That should lock up the thing for about a minute. It's busy. The television set at home is grinding away, going, wait a minute, I'm trying to stream video. Why are you calculating on me right now? It's going to draw another plot, look about the same. So here's what happens if we do 5,000 simulations. We still have some non-zero values less than 10% and some non-zero bigger than 10%. But with enough simulations, it's clear that most of the defects will hover around this 10% rate as expected. So if we did a measurement and calculated a defect rate of, say, 0.2, according to this picture, we would conclude that is not in our design rate. There's something wrong. It's time to maintain the machines. Or we can do it this way and have all sorts of numbers to throw back at management to justify why we shut the line down for 2 days and fix the broken machinery. And that is one main use of this kind of testing. Let's go to the next one. I have to go through this sort of quickly because I babbled aimlessly for some reason. A/B testing. In modern data analytics, deciding whether two numerical samples come from the same distribution is called A/B testing. And the name refers to the labels of the two samples, A and B. If you have a third one, it's C, which is easy as 1, 2, 3. A, B, C, easy. Sorry, little Michael Jackson there. So imagine you're in charge of a website, say an online video game shop, if there is such a thing anymore. And you have a current version of the website, call that A, but you don't like it. For instance, you're not making as much money as you want. And so you're going to propose to change the design of the Add to Cart button. And so you'll call that website with a different button B. So there's A and there's B. And you want to see if maybe that will increase your sales. You probably should have a thing C, which is to get the customer to enter the credit card information first and then start stealing while they're going through the website. But you don't do that yet. You're still honest. You haven't been Googled or Appled or Facebooked sufficiently yet. So button A looks like that. Button B looks like that. And the question we want to ask is there a difference in recorded sales based on which button is provided to the customer? And I know that sounds dumb to us, but there are actually people that make a living asking questions like that. It's good work if you can get it. I wish you all do. But you will eat your arm off to get away from the boss at some point. So you set up the website and you design it so that when people hit the landing page, about half of them are sent to the old website, A, and the other half to the new website, B, which differs from A by a button. This is called an A/B test. And you have data from both and you want to know, does the change I made increase the sales? We're not looking about causality. We don't care if the button made a damn bit of difference or not. But if the one with button B generated substantially more sales, what do you think management's going to tell you to do the website? I'm going to say switch to button B because the whole point is to make money. So why not just change the website and monitor it for a week? That's a fair question. By having two sites alive at once and randomly directing users, you have controlled for all the other possible variables. But if you do 1 week and then a different week, if you happen to choose the week just before Christmas, Christmas itself is going to impact the sales, especially this Christmas coming up, which is due to supply chain problems. Shipping will be delayed indefinitely, but we will take your money and store it for you. So you want to run them in certain instances side by side. This is a case where you want to do that. You want to remove the time of year having any impact on your decision making. And then why is it not called an ABC test? Well, that's because we don't have residuals from Sony to do ABC. Easy as 1, 2, 3. You can do as many perturbations as you want, but you need to keep the name simple. If you do more than 1 or 2 changes, you start reducing the number of sample size in each one. So if you're Amazon, I mean, you get a couple billion people a day. You can have all different website designs that you can imagine, and you'd be guaranteed at least getting 10,000 on each of them. But if it's the rest of us, that maybe we get 300 people a day, if we have 6 different changes, you're looking at 50 customers per change. That's not a very big sample size. We've already seen up here what happens to that when the sample size isn't too big. There's a lot more graininess in the results. So generally, it's just a few changes at a time. So let's assume we have 1,000 people, and our machine sent 550 to site A, 450 to site B. We would go back to our programmer and say, hey, what's this? It's supposed to be half and half, but it says, sorry, your contract's done. You have to live with it. So at site A, 48 of those 550 bought something, and in the other button, 56 out of 450 bought something. We want to ask the result, are those differences significant? And so first, let's just calculate the purchase rates. So the purchase rate of A is about 8% of the people that visited bought something from site A, and B, 12% bought something. So at first glance, it expresses a rate. Yeah, B has almost went from 8 to 12. So that's like almost 50% more in B. We might be tempted to say, well, B is obviously better. Let's just switch now. So this happens to be a binomial type distribution, but because we are diligent website interface engineers and we want to give our client good value, we're going to produce a plot. And so there's our A group and B group plotted. Almost the same color choice here. That's purely coincidence. That would be cool if I planned it. And we see there's a difference in the plot, but there's also a fair amount of overlap. And so our reasonable question is that difference real, or could it be explained by randomness? And that's just like the machine tool shutdown decision. We want that area to be smaller than some value. So we would conclude here, I would conclude that B has an edge, but it's quite possible that if we picked any 2 random points, we wouldn't know which histogram it came from. So we're interested in the average number of sales. It turns out if we do that, averaging the underlying distribution means that our final estimate will also be normal distribution. That's a nice feature of that. And so we can do things in terms of averages and then make some distribution plots, and that gives us a better way of assessing our type 1 error. Now we still want to ask ourselves if B is statistically significant. So one thing we can do is build a script that will tell us what the z-score is and the associated p-value of this overlap area. So we'll do that again using all the same original data. We get this cool graphic, and the area associated with the difference in purchase rates starting from 0 working to the right is 97% of the whole area of, I think it's based on A. So we can say that given the null hypothesis, B is less than or equal to A is true, we would expect to get that picture 97% of the time, and a more extreme result only 2% of the time. So now we've made a lot of plots to try to do this. Let's instead try to calculate p-values. So we're going to create a function called getConfidence. We're going to keep the clicks on A, the number of A, clicks on B, number of B. So that's going to produce the two sales rates. Then we're going to calculate a z-score of the difference between the two rates. If that z-score is small, close to 0, there's no difference. If the z-score is big, there's a difference, and we declare the level of bigness by our choice of alpha. And so here's our getConfidence test. All it's going to do is return the z-score. Now we can use our parametric or non-parametric hypothesis test. We'll just use Mann-Whitney, which is non-parametric because we don't have to test for normality. And we run that thing, and it says the null hypothesis of B being less than or equal to A, the probability of that being true is 2%. So if the probability of B being less than A is 2%, the probability of B being bigger than A is 98%. And our goal is B bigger than A, so we have pretty good evidence in this particular example that B is a reasonable change to make. You can learn more if you go read these references. We will start lab in 15 minutes, and today's lab, and there's also homework, today's lab is just playing with these ideas. You don't want to look at the experimental work for future semesters because that obviously has no relevance today. So I will see everybody in 16 minutes or so. Go find a nice comfortable place to enjoy lab. And hopefully Josh got my email last night and has had a chance to look at it. Otherwise he's going to be very confused. Okay, so shut down the recorder.